I'm happy to be able to introduce our seminar speaker today, Dr. Priyanga Amarasekare. Priyanga is originally from Sri Lanka, where she completed her bachelor's degree in zoology. She has a master's degree in zoology also from University of Hawaii at Manoa and a PhD from UC Irvine. She was an assistant, she was a postdoc at the National Center for Ecological Analysis and Synthesis, and then her first academic position was at University of Chicago. She moved from there as an associate professor to UCLA, where she continues as professor now. Her earliest research, I think, was in the area of conservation, and that's really continued as a theme throughout her career, the action orientation of her science. Her work became increasingly mathematical and theory-laden, beginning with her PhD. And again, that theme really characterizes her research now. Her work is primarily in population and community ecology, and she's made major advances to understanding how environmental variation, environmental change, especially variation and change in space, affect the whole range of issues from populations to species interactions to communities, and food webs now, I guess. She's been widely recognized for the quality of her research and the impact of her research. Her academic honors begin with the major early career award from the American Society of Naturalists, the Young Investigator Award, and she very recently received the Ecological Society of America's highest mid-career research award, the MacArthur Award. And she has many others in between those as well. She also has been impactful in areas beyond research. She is the recipient of a Distinguished Teaching Award from her work at UCLA. She also has contributed significant professional service. She's currently the vice president of the American Society of Naturalists. You could say a lot about Priyanga's work, but I want to leave most of the time for her. So I'm going to read a few sentences from her MacArthur Award citation because I think it really nicely describes why her work is important. Dr. Amarasekare is a rare ecologist whose work is simultaneously broad and deep and has important effects on every area it touches, from coexistence theory to climate change. Her distinction derives from making fundamental advances in the field while simultaneously tackling and solving important practical problems, using an integrative approach that strikes the balance between mathematical tractability, biological realism, and societal importance. Like Robert MacArthur, she has demonstrated exceptional ability in identifying maximally simple, yet not simplistic models to achieve new insights on important ecological problems. Today, she'll speak to us on predicting the effects of climate warming from chemistry to evolution. Priyanga?
You can hear me now. Can you hear me? Yes, well, thank you for the wonderful introduction. I had to remind myself to read that citation when I'm feeling bad about myself. I want to actually, it's been a great pleasure being here. I've had a lot of fun talking to a lot of you and learning a lot of things about your research. And I want to thank the graduate students once more for inviting me and giving me this opportunity to talk to you about my work. So my work, very broadly speaking, is focused on the interplay between how the interplay between biotic interactions and abiotic variation influences ecological and evolutionary dynamics. Now, I go about this, let me just go back. I go about this using what I call my three-step process. So the first step is to determine how biotic and abiotic factors affect fitness through their effects on phenotypic traits. The second step is to determine how fitness effects translate into ecological processes such as population regulation and species coexistence. The third step is to determine how these ecological processes feed back into the trait response, causing trait evolution. So my goals with this work are to explain the patterns of dynamics and diversity we see in nature and to predict how these patterns change due to perturbations to organisms' typical abiotic and biotic environment. My approach is a combination of observations, experiments, and mathematical models. I try to develop general theory that applies across taxa, habitats, geographic location, and which provide the conceptual underpinnings for addressing environmental problems such as conservation, invasive species, and climate warming. Now, so within sort of this very broad theme, I've done a lot of different things. And this is just an example. You don't have to actually read all of that, but it's just to show that I worked on a number of different things. I'm interested in a number of different things, can really actually focus on one thing. But today I want to focus in particular on one topic, which is about the fact that organisms are we live in a world where organisms are continually being exposed to novel environments. And I want to make the argument that predicting how organisms respond to novel environments requires developing theory that integrates across levels of information from chemistry to evolution. So I focus on temperature variation and phenotypic plasticity, not least because temperature is integral to all life processes, and climate warming poses one of the greatest threats to life on Earth. Now, organisms use phenotypic plasticity as a strategy to maximize fitness in variable environments. Plasticity is manifested as a reaction norm, which is the range of phenotypic responses exhibited by a given genotype in response to environmental variation. Now, predicting the effects of climate warming requires knowing whether organisms have sufficient plasticity to respond to current levels of warming and whether the plasticity can evolve fast enough to keep pace with warming. Now, but we know that existing plasticity is the result of past evolution, while future plasticity is the result of evolution that's yet to occur. My thesis is that chemistry is integral to understanding both past evolution and predicting future evolution. Now, we know that evolution results from the interplay between selection and constraints. Selection occurs when biotic and abiotic factors generate variance in fitness. Constraints are factors that prevent organisms from maximizing fitness, even when they may have a selective advantage. So here's my argument. Temperature imposes constraints on biochemical reactions that are integral to life. These biochemical constraints, in turn, limit the set of possible ecological and evolutionary outcomes. I'm going to start by showing you 4 examples of temperature-imposed constraints on biochemical reactions. Data show that the mutation rate increases with temperature in both prokaryotes and eukaryotes. Higher temperature increase mutations with phenotypic effects. Now, we know that most mutations are deleterious. This means that rising temperatures could lead to the accumulation of deleterious mutations as we observe this sort of ongoing warming. Data also show that temperature can limit transcriptional fidelity. Transcriptional errors can affect the structural integrity of proteins, and errors that evade quality control can disrupt eukaryotic cell physiology. Higher temperature, of course, higher temperatures mean more errors and greater disruption of protein function. It has long been known that temperature drives enzyme activity with a faster rate of decline at high temperature extremes compared to low temperature extremes. Data show that above a low temperature threshold, mortality rate of all ectotherms increases with increasing temperature. This is because all reaction rates increase with increasing temperature. So it's a case of living faster and dying sooner. So temperature imposes a fundamental constraint on longevity. So we see that temperature can impose constraints on biochemical reactions that are integral to life. There are other examples. I wanted to show these 4 representative ones. So the next step is to determine how these biochemical constraints limit the set of possible ecological and evolutionary outcomes. Now, biochemical constraints limit ecological outcomes by imposing constraints on phenotypic plasticity. They limit evolutionary outcomes by imposing genetic and demographic constraints. So I'm going to talk about biochemical constraints on phenotypic plasticity in the context of thermal reaction norms of ectotherms. These ideas mainly apply to animal ectotherms. I'm really happy to work on plants if somebody wants to help me do it. So animal ectotherms exhibit 2 types of thermal reaction norms. One type is unimodal and symmetric with similar rates of decline at low and high temperature extremes. Reproductive and consumption traits tend to exhibit this kind of response. The second type is unimodal and left-skewed with a faster decline at high temperature extremes. Maturation and many performance traits tend to exhibit this kind of response. So looking at some data, these are the data for the maturation, for development for a large number of arthropod species. You can see that the maturation rate exhibits a left-skewed response. These are the data for performance traits. We have enzyme activity in lizards, tongue flicking and digestion rate in snakes, and endurance and burst speed in lizards. They all show a left-skewed response. And here we have the data for mortality, which was a monotonic response above a low temperature threshold, again for large numbers of different species. And here we have data for reproductive traits, again for a large number of different species. You can see that they tend to exhibit a symmetric unimodal response. And finally, here are the data for attack and maximal uptake rates of ectotherms. They both tend to exhibit a symmetric unimodal response. That's for a large number of species, from invertebrates to reptiles. So what we see is that the qualitative nature of thermal reaction norms appear to be conserved across ectotherm taxa. This is encouraging because it allows us to develop a trait-based framework for understanding temperature effects that can apply broadly across taxa. Now, the first step is to understand the difference between these 2 types of thermal reaction norms. We do this using reaction rate theory. So this is the Boltzmann-Arrhenius function, where Kt is the trait value at some temperature in Kelvin, Ktr is the trait value at a reference temperature, and A sub k is the Arrhenius constant, which quantifies the temperature sensitivity of the trait, meaning how fast or slowly it changes with temperature. Here is a modified reaction rate function that incorporates the reduction in reaction rates at low and high temperature extremes due to enzyme inactivation. So let's focus on the denominator of this equation. When the magnitude of AH exceeds the magnitude of AL, we get a faster decline at high temperature extremes. In this case, the reaction is rate controlled. It increases to a maximum, followed by a rapid decline. When the magnitude of AH is approximately equal to the magnitude of AL, we get similar declines at low and high temperature extremes. In this case, the reaction is regulated. It's prevented from increasing to its maximum. So this difference between rate-controlled and regulatory responses is really important. Rate-controlled responses are under very strong biochemical control. Reactions tend to proceed to their maxima, followed by a rapid decline. Regulatory responses are driven by negative feedback processes that prevent these reactions from proceeding to their maxima. So This suggests that the rate control reaction norms may be subject to different types of constraints at low and high temperature extremes. Data support this idea. The decline at low temperatures occur through via phenomena such as the freezing of body fluids, which occur relatively slowly. The decline at high temperatures occurs via protein denaturation and loss of respiratory function, which occur very rapidly. So the key point is that rate control responses are subject to stronger biochemical constraints at high temperature extremes. So we see that biochemical constraints can impose limits to phenotypic plasticity. The next step is to determine how these limits in turn dictate ecological outcomes. And I'm going to talk about this in the context of climate warming. So climate warming means exposure to warmer thermal regimes, making it more likely that organisms will reach the upper limit of thermal plasticity. We expect rate control responses to be more adversely affected by warming than regulatory responses. So to make this more concrete, let's look at the thermal reaction norms of life history traits. So here we have the temperature responses of birth, maturation, and mortality rates under typical seasonal variation for a temperate species. The shaded region depicts the typical temperature range experienced by the species. So here, the top graph is for typical seasonal variation you just saw. The bottom graph depicts trait responses under warming. The pink and purple areas denote the new thermal regime. So we see that warming tends to reduce the birth rate. But the strongest effects of warming are seen in maturation and mortality rates. There's a steep decline in the maturation rate and a sharp increase in the mortality rate once temperatures exceed the typical upper limit. So we see that rate control responses such as maturation and mortality are more adversely affected by warming than regulatory responses such as birth and attack rates. So the next step is to determine how these warming effects on thermal reaction norms translate into ecological outcomes. I'm going to do this in the context of warming effects on phenology, the seasonal timing of life history events. Now, it's well known that warmer winters cause an advance in phenological events. Flowers bloom earlier, insects emerge earlier. But if winters are too warm, summers are necessarily going to be too hot. But it turns out we know relatively little about what happens during these hotter summers. In order to find out, we have to determine how biochemical constraints on thermal reaction norms translate into phenological changes at the population level. We do this using trait-based models of population dynamics. Now, so here is a simple depiction of an ectotherm life cycle with juveniles and adults. Adults produce juveniles, juveniles develop into adults, and you have mortality at each stage. Here's a mathematical depiction of that stage-structured life cycle using delay differential equations. So here is the equation for the juvenile stage. Here is the equation for the adult stage. Recruitment to the juvenile stage occurs via adult reproduction. Losses occur via maturation and mortality. Recruitment to the adult stage occurs via juvenile maturation. Losses occur via mortality. Density dependence can operate through fecundity, juvenile mortality, or adult mortality. This is the maturation function that incorporates the temperature-dependent developmental delays that characterize ectotherm life cycles. This function is critical in predicting phenological shifts due to climate warming. So because it's so important, oops, wait, did I get this right? Yeah. Let's look more closely at this maturation function. So maturation of juveniles to adults is a function of the birth rate some tau time units ago, times the rate at which individuals develop from juvenile to adult, and survivorship during juvenile development. It's clear, right? So there was some adults lay some time ago, they develop into juveniles, and some die, some survive to adulthood, right? That's all just depicted here. So now here's the important thing. When temperature varies over time, developmental delay and juvenile survivorship vary with both temperature and time. So we see that because of this dependence of both temperature and time, maturation and mortality tend to play a key role in ectotherm population dynamics. Okay, so here is the full model. And I'm going to analyze this model on the typical seasonal variation and warming. Let's first look at population dynamics under typical seasonal variation for an insect species inhabiting a Mediterranean climate. So here is the typical phenological pattern over the year under typical seasonal variation. Note that there are two peaks in abundance, one in the spring and one in late summer. So here is the phenological pattern when there's a 3-degree increase in the mean annual temperature over a period of 100 degrees. So the black curve here is the typical phenological pattern. The blue curve is the pattern after 100 years of warming. So you can see that the spring peak has advanced, but the summer peak is delayed. This is because warmer summer temperatures reduce birth and maturation rates and increase mortality, leading to a decline in abundance. And that causes a delay in the subsequent increase in abundance. Here's the phenological pattern when there's a 5-degree increase in the mean annual temperature. So the spring peak has advanced, but the summer temperatures are so warm that birth and maturation cease and mortality increases rapidly, leading to a very large decline in summer abundance. Notice that now population growth is now restricted to early spring and autumn. Now we see this pattern precisely because warming has more adverse effects on rate control traits. Hotter summers lead to a steep decline in the maturation rates and large increase in mortality rate, leading to a large decline in summer abundance. The hotter the summer, the more difficult it is for species to recover from this decline in abundance. Now, this summer decline is exactly the pattern observed in disease vectors and pest-enemy interactions. Warming affects vector-borne diseases like West Nile, malaria, dengue, through its effects on vector phenology. Hotter summers change vector abundance patterns from a single summer peak to a bimodal distribution with spring and autumn peaks. This reduction in summer abundance, if sufficiently large, can cause disease extinction from warmer climates. And in fact, that's predicted to happen for malaria in West Africa. So we see, and we've seen an example where biochemical constraints on phenotypic plasticity can dictate ecological outcomes. Next, we want to determine how biochemical constraints limit evolutionary outcomes through enforcing genetic and demographic constraints. So there are two parts to this question, and I'm going to investigate each one in turn. So let's first look at how biochemical constraints impose genetic constraints. Plasticity can evolve in response to warming if there's heritable genetic variation. So availability of heritable variation imposes a genetic constraint on the evolution of plasticity. Importantly, this genetic constraint arises from arises from biochemical constraints on existing plasticity. So to see this, recall that rate control responses are under stronger biochemical constraints than regulatory responses. This means that rate control responses are subject to very strong stabilizing selection. As a result, they tend to exhibit very little standing genetic variation. So that's a hypothesis we can test by quantifying genetic variation in thermal reaction norms. So we did this thing using the harlequin bug, which is a hemipteran herbivore feeding on an endemic plant species in the California coastal sage-grouse. This is one of the very few instances in which genetic variation in thermal reaction norms have been quantified for a species living in the wild. So here are the data for genetic variation in rate-controlled and regulatory reaction norms from our experiments. On the left, you have data for the maturation rate. On the right, you have data for the birth rate. Each curve represents a distinct genetic line. You can see that the rate control reaction norm exhibits very little standing genetic variation compared to the regulatory reaction norm. So we saw before that responses to current levels of warming are limited by strong biochemical constraints on rate-controlled reaction norms at high temperature extremes. What these genetic data show is that responses to future warming are likely to be limited by strong genetic constraints on the evolution of rate-controlled reaction norms. Okay, so we've seen that biochemical constraints can limit evolutionary outcomes by imposing genetic constraints. Now I want to show you that biochemical constraints can also limit evolutionary outcomes by imposing demographic constraints. I'm going to illustrate this with an interesting biogeographic pattern. Data show that tropical ectotherms are more successful in invading temperate habitat communities than vice versa. Some of the most strongest evidence for this comes from paleontological and biogeographic data. What this work shows is that tracks are originating in the tropics and expanding the distribution, that diversity at higher latitudes primarily result from taxa originating in the tropics and expanding their distributions. Taxa originating at higher latitudes and expanding the tropics are rare, as are the reinvasion of the tropics by higher latitude taxa. Data on contemporary invasions show much the same pattern. The most successful invaders of temperate habitats are ectotherms of tropical origin. Some of the most striking examples involve the cane toad, the Asian tiger mosquito, fire ants, scale insects, and marine invertebrates. In contrast, the most successful temperate invaders of tropical habitats are endotherms, such as passerine birds and rodents. So we see a directionality in adaptation to novel thermal environments. Tropical species can adapt to temperate environments, but temperate species cannot seem to adapt to tropical environments. So the key question is, why do we see such a pattern? To answer that question, we need to understand how thermal reaction norms evolve in novel thermal environments. I'm going to do this with an eco-evolutionary framework that combines the quantitative genetics of reaction norm evolution with stage-structured population dynamics. I'm going to focus on the evolution of the thermal reaction norm for the birth rate because it's a key component of fitness. It's also a regulatory trait with standing genetic variation, so there is no genetic constraint to evolution. But it evolves against the backdrop of maturation and mortality rates, which are rate-controlled reaction norms subject to strong genetic constraints. So ecological dynamics are given by a stage-structured model, very similar to what you saw before for the phenological effects. Now we saw before that biochemical constraints on rate control responses can cause a large decline in summer abundance, right? When we talked about the phenology before. Now, the important consequence of this is that it can predispose populations to extinction while demographic stochasticity during periods of low abundances. This is the demographic constraint that biochemistry imposes on reaction norm evolution. So the feedback between ecological dynamics and reaction norm evolution is given by the mean fitness of the population. This is a function of two important quantities. The first one is the mean reaction norm, where X bar is the mean value of the quantitative trait that drives the evolution of the birth rate. The second is the density-dependent effects on life history traits that drive population regulation. So here we have the full model with ecological dynamics, with stage-structured population dynamics given by these two equations you saw before, and trait evolution given by this third equation, where H squared is the narrow sense heritability, sigma squared is the phenotypic variance, and the partial derivative gives the selection gradient, which is the rate at which mean fitness changes with changes in the mean trait value. Now, I'm going to use this model to investigate the two scenarios we are interested in. I'm going to do this by parameterizing the model with trait response data from two hemipteran insect species from tropical and temperate latitudes. The tropical species is a pod-sucking bug from Benin, which experiences a thermal regime characterized by high mean temperature and low amplitude fluctuations, tropical. The temperate species is the pea aphid from York, England, which experiences a thermal regime characterized by a low mean and high amplitude fluctuations. Right. So here we have the observed thermal reaction norms for the birth rate for the tropical and temperate species. In these graphs, the black line is the mean habitat temperature. The red line is the optimal temperature for reproduction, and the dashed lines give the range of seasonal variation experienced by each species. Now, because both species are native to the habitats they reside in, it's reasonable to expect that natural selection has shaped these reaction norms so as to maximize the species' fitness in its typical thermal environment. So what I'm going to do is to compare these observed thermal reaction norms with the ones that evolve in the model. Right. So let's start with the temperate species and see what happens when we subject the temperate species to its typical thermal environment. So here, the red curve is the empirically observed thermal reaction norm for the birth rate. The blue curve is the reaction norm that evolves in the model. You can see when the temperate species experiences its typical thermal environment, its reaction norm evolves to resemble the one that's observed in nature. Right, so now let's see what happens when the temperate species invades a tropical climate. Now something different happens. Exposure to high tropical temperatures cause abundances to fall so low that stochastic extinction occurs before the species has the opportunity to adapt to a tropical thermal environment. All right, so let's now look at the tropical species, starting with its typical thermal environment. And here, again, the red curve is the observed reaction norm, empirically observed reaction norm. The blue curve is the reaction norm that evolves in the model. We can see when the tropical species experiences its typical environment, its reaction norm evolves to resemble its empirical pattern. All right, now let's see what happens when the tropical species invades a temperate climate. So here, The green curve is the empirically observed reaction norm in a tropical habitat. The red curve is the empirically observed reaction norm in a temperate habitat. And the blue curve is the reaction norm that evolves in the model. So we can see when a tropical species is subject to a temperate thermal regime, it evolves a reaction norm that's characteristic of a temperate species. So we see that when a tropical ectotherm invades a temperate habitat, its reaction norm rapidly adapts to a temperate thermal regime. But when a temperate ectotherm invades a tropical habitat, stochastic extinction during the initial invasion phase precludes adaptation to a tropical thermal regime. Now, let me show you how this happens, starting with the temperate species. So in these top graphs, the red curve is the pattern of seasonal variation over the year. The black curves are maturation and mortality rates over the year. The bottom graph gives abundance. So here's the key point. When a temperate species experiences a tropical thermal environment, thermal reaction norms of maturation and mortality reach the upper limit of plasticity. The maturation rate is the lowest and mortality is the highest during the long period of warmer temperatures. As a result, abundances fall to the low levels at which the population is predisposed to extinction via demographic stochasticity, resulting in the inability to adapt to this harsher thermal environment. Now let's look at what happens in the tropicals. When the tropical species invades a temperate habitat, it can take advantage of the warmer periods of the year. So maturation rate is highest during this time, which will be summer. And although mortality is also high, it's much lower than what a tropical species experiences in its typical thermal environment. As a result, abundances do not fall to the levels at which stochastic extinction would occur. And as a result, the species can persist long enough to adapt to a temperate thermal environment. So here's a case where there is strong selection, there is heritable genetic variation, but evolution is prevented by the demographic constraint of extinction during low abundances. So to summarize these results, we know that rate-controlled reaction norms are subject to stronger biochemical constraints at high temperature extremes. These biochemical constraints impose a demographic constraint that precludes adaptation to a warmer thermal regime. The result is a largely reversible biogeographic pattern. With that, we come back to the argument I made at the beginning, which is that temperature imposes constraints on biochemical reactions that are integral to life. And these biochemical constraints, in turn, limit the set of possible ecological and evolutionary outcomes. Now, as I noted at the beginning, predicting the effects of climate warming requires knowing whether organisms have sufficient plasticity to respond to current levels of warming and whether their plasticity can evolve fast enough to keep pace with the rapid pace of warming. What we find is that responses to current levels of warming are limited by strong biochemical constraints on rate-controlled reaction norms at high temperature extremes, while response to future warming are limited by demographic constraints imposed by these rate-controlled rates and strong genetic constraints on the evolution of rate-controlled reaction norms. And hence my argument that predicting the effects of climate warming requires developing theory that integrates across levels of information from chemistry to evolution. Oops. Well, actually, for all of those nerds out there. We know that so there's a physical pattern where a hot object cools down spontaneously, right? But it takes work to make a cold object hot, right? So we've just seen a biogeographic pattern where moving from hot to cold, because why does it work like that? Because everything works in the direction of increasing entropy, right? So we've just seen a biogeographic pattern where moving from hot to cold is likely, where it's involves an increase in entropy, but moving from cold to hot, which requires work, seems unlikely. So there is some connection here that I haven't yet worked out, but it seems really interesting that you would see the same pattern at this level that you would see, just the everyday physical pattern. Anyway, with that, we have a number of challenges for future directions with theory. One of these is that we really need to go beyond the standard reaction kinetics theory, reaction metabolic theory, to incorporate how feedback processes arising from things like neuron and hormonal regulation interact with enzyme kinetics to regulate these trait responses. And a second thing is to incorporate, and I really think this is very important if you want to make progress, is to incorporate mechanistic descriptions of molecular level processes into models of ecology and evolution. And with that, I want to thank my collaborators, especially these amazing undergraduates. Where's my pointer? Who helped me with the experiments, my funding organizations, and thank you all for coming and listening.
Okay, we have time for questions, and I want to point out